Hello, everyone. Thank you for coming today. My name is Erika De Benedetti, and I will manage this webinar. Before to get started, I would like to give you a couple of information. Firstly, this webinar is recorded, and the recording will be available on biostimulant.com website and on our social media in the next days. Secondly, I invite you to use the Q&A box at the bottom of your screen to submit questions. We will collect your questions and answer to at least some of them at the end of the presentation. The topic of today is biostimulants and plant hormones. It takes two to tango. It's my pleasure to introduce our speaker, Danny Geleen, professor in the Department of Plants and Crops, Faculty of Bioscience Engineering at Ghent University. Welcome, Professor. The floor is yours.
So, thank you very much, Erika, for this kind introduction. So I'm Danny Geleen, and I'm leading the lab unit Horticell, and where we do a lot of research on biostimulants and plant hormones. So many of you know that biostimulants come in very many different types and flavors, and that in many cases we have been trying to categorize these biostimulants according to where they come from or what kind of composition they have. And so we distinguish, in this case, 5 different groups, which I define as extracts, for example, from seaweed, plant or microbial origin, or organic waste. Another group that is very common, known in the biostimulant field, is acids, and there are many kinds of acids. You have these humic acid and fulvic acids that are derived from partial degradation of plant material, but you have also many biostimulants that are amino acids, organic acids, and even hormones can be considered in many cases to be acids because they have acid site groups. Another category that's very important are the hydrolysates, which are basically fermented products that are derived from animal waste, plant waste, or even microbial waste. And then we have, let's say, a more diverse group of biostimulants that can be classified as other chemicals, and these contain antioxidants, oligo or polysaccharides, adsorbents like biochar, etc. And finally, a little bit on the side are the microbes. They are genuine biostimulants because also they are being recognized by the European legislator as biostimulant products. And here we know that there are beneficial fungi and bacteria that can be applied that have promotional effects on plants. And so it's very important here to state is that fertilizers as such can stimulate plant growth, but they are not considered as biostimulants. They should be seen as fertilizers and only as fertilizers. Now, while this categorization based on content or type of product can be very useful and is still in many reviews used, that categorization or grouping does not tell us much about the bioactivity. In fact, the different groups that we just depicted, they cannot be directly linked with the different activities that we wish to achieve. So here I've tried to categorize biostimulants based on what kind of activity they have. And there are many types of activities that we wish for. For example, we want to have a specific fruit size, we want to have increased or decreased shoot and biomass or root biomass, we want to promote germination, we want to have a better root architecture, etc. These type of processes that we can influence, I would categorize them under growth. A second group of beneficial impacts that we report for biostimulants are related to metabolism. And here I would include, for example, promotion of photosynthesis, fruit or in general crop quality, content in metabolites, etc. But also the capacity to take minerals from soil or other environments under conditions of biostimulant treatment, and even so the influence of biostimulants on the microbiome, be it in the root system or even at the leaf level. And then a third category, very common for plant biostimulants, is to mitigate stress. And here we see a number of external factors that we wish to mitigate. We have the abiotic factors like heat, stress, drought, salt stress, flooding, anoxia, cold, wounding, but even flowering time, which we know is influenced by stress, is an important factor that we want to regulate using biostimulants. And here an important exception is that none of these activities can be related to biotic stresses, so no pathogens or diseases should be one of the targets for biostimulants, because that is specifically only for biopesticides. So when we look at these three groups of activities, then we can try to understand how biostimulants may be working. So here I've tried to categorize the three groups again, but now try to show what kind of underlying processes may be important for these categories. When we look at growth, it is clear that plant growth is primarily controlled by cell division and cell expansion. There is no other way that plants can increase in size by modulating either of these two processes. A metabolism can be categorized as primary and secondary, and so we see that biostimulants can have impact on both of these, or specifically on one of them. And then when we look at stress response, then biostimulants can contribute to damage repair, or they can also contribute to protection for future damage that may be there when another stress moment appears, and that plants are then better prepared for this secondary stress effect. So how are these processes controlled? Well, cell division and cell expansion are known to be controlled by development, depending on stage of your plant life position or your life cycle. You will have different developmental stages, and that will affect cell division and expansion. Organ initiation will also be an important process. And then processes like dormancy will determine whether cell division or expansion can take place, and this is then controlled by external factors, for example. When we look at metabolism, there we see that primary and secondary metabolism involve energy metabolism, so photosynthesis will provide you the necessary energy molecules that can drive primary and secondary metabolism. We have metabolite synthesis, we have catabolism, so breakdown of metabolites, but also the storage and transport will determine to what extent we have a specific metabolism. When we look at stress response, here we see that damage repair includes, for example, protein degradation, degradation of damaged lipids. DNA does not get degraded, but there you have repair directly with specific enzymes. And then we have, during stress response, in many cases, the production of radical oxygen species, or in short, ROS. And this can, in turn, also cause damage and needs to be mitigated, and can be mitigated by, for example, biostimulants. Osmolyte synthesis, etc., can also be important for future protection. Now, many of these processes are, in fact, also controlled by plant hormones, which is largely the topic of today. And what we see is that cell division and cell expansion are controlled by many different hormones. Cell division expansion is primarily controlled by auxin and cytokinin, but for example, gibberellic acid can stimulate cell expansion, like steroids, trigalactones will, for example, stimulate the outgrowth of shoots. So these these hormones, excuse me, are very important in controlling cell division and cell expansion. When we look at metabolism, there we have primarily gibberellic acid as a very important regulator, but equally other hormones. There are reports that show cytokinin and auxin to be involved in regulating the metabolism. And stress response is probably the most diversely controlled process That includes many different hormones. Nearly all the hormones that we have studied so far do play a role in stress response. And here we have abscisic acid, ethylene, gestanoic acid, salicylic acid, polyamines, and auxin all have been shown to be involved in stress response. So let's first have a look at control of cell division. Here we see the mitotic cycle, which is depicting the process that you need to create a divided cell. And basically you go through 4 different stages that determine whether a cell will go into mitosis and divide. So the first step is the G1 stage that prepares the cell for duplicating its DNA during the S phase, which is then followed by a preparatory phase for mitosis, during which the cells will segregate chromosomes and create 2 new cells. It's clear that when you increase the cell number, that you will increase growth. There are, however, different types of cycles, and one of them is the endocycle. And here the step of mitosis is omitted. So basically you have a preparatory G phase followed by a DNA synthesis phase, but there is no real mitosis. So basically what happens in this situation is that your cell is duplicating its DNA. Cells will then enlarge, but they will not segregate chromosomes. And here we see that this process is often linked or associated with cellular expansion and also cell differentiation. So following these 2 different cycles, we can get more cells or we can get larger cells, and those increase in cells after cell division. They can also, of course, contribute by having additional cell expansion. Cell expansion is a very different process that does not involve DNA synthesis, does not involve the multiplication of cells. However, what happens here is that the original cell will actually change properties of its cell wall. It will loosen up the different structures within a cell wall, including the cellulose with its microfibrils that are connected among each other through pectin and hemicellulose. And by loosening up these structures, the cell wall gets a bit weaker. Upon increased turgor pressure or maintaining a certain level of turgor pressure, then the weakened cell wall will allow the expansion of the cell to take place. And this can contribute to a large extent to increase in biomass. So we've got these 2 processes. And if we want to understand how a biostimulant works, we need to understand whether it stimulates the cell division or cell expansion, or perhaps both. So when we look at the control of cell division, that is mediated by means of specific proteins, a protein complex that we know as the cyclin-dependent kinases that bind to cyclins. And different complexes of CDKs and cyclins will allow the transition of the different cell cycle phases. For example, here we can see that a CDKA linked to cyclin D will stimulate the transition from the G1 phase into the DNA synthesis phase. And so these transitions are mediated by different combinations of CDKs and cyclin. So basically what we understand from this is that if you want the cells to divide, you need to activate these different CDKs and cyclins. And this can be done through a developmental program and internal stimuli, but also by means of hormones. Hormone signaling has been shown to modulate the CDKs, and cyclin proteins increase their level by means of transcription, for example. However, these same regulators are also controlled by, for example, abiotic stress. And here we have an intermediate regulatory system. It involves, for example, calcium kinases, ROS signaling that influence the transition of these different steps. And usually they actually inhibit the activation or inhibit the activity of the CDKs and cyclins. If we look in a bit more detail, then we summarize the different publications in this field of research. And what we can see is that, for example, a hormone like auxin and energy from sucrose together can promote the transition from the cycle G2 to the mitotic phase. So this is kind of interesting. By applying the hormone auxin, you can accelerate the transition from G2 to M. Likewise, this is achieved by CDK. So this is an important step in the decision whether a cell will fully commit a mitotic division. And in some cases, we see that cells are being blocked at this stage because they are not allowed to progress and commit a full cell division. The entry of cell division is also stimulated by cytokinin through a different group of cyclins. Cyclin D, for example, this family of proteins is being activated by the application of cytokinin. And here we see that this stimulates the G1 to S checkpoint. So when we apply cytokinin to plant cells, tissues, especially in vitro, we see that we can promote cell divisions. And often it's a combination of an appropriate level of cytokinin with auxin that can stimulate cell divisions to such a dramatic rate that, in fact, the cells start to form callus when they're grown in tissue culture conditions. Now, these are not the only hormones that seem to be influencing the cell cycle. So there is, for example, stimulation of the G2M checkpoint by gibberellic acid. Ethylene seems to be inhibiting this transition. And then we have a bunch of hormones that actually can also help the cells to go out of the cycle and stop cell division altogether. And for example, jasmonate application will tell the cell maybe it's better to wait for a while before you go into another round of cell division. Likewise, ethylene and ABA stimulate the activation, what we call the G0 phase. And this is a status of cell that is latent, which is preventing the cell from continuing to divide. And so we have these different hormones balancing each other out. For example, here we have ABA that will activate CDK kinase inhibitors, which then will block the CDKs and therefore will prevent ultimately the checkpoint to be transitioned by this group of CDK cyclin Ds. So basically, even if under conditions where you have applied cytokinin, but the cell division conditions are not favorable because of maybe external factors that should prevent it, ABA can overrule, for example, the activation of this transition. So you can see that the hormonal regulation of the cell cycle is rather complex, and that will clearly have an impact on whether a biostimulant will be able to influence cell divisions or not. To make it a bit even more complicated, when we look at how hormones are actually operating in plants, we need to understand a little bit about hormone homeostasis. Basically, what happens in plants is that they will regulate cell division and cell expansion processes by producing hormones, and so this will belong to the category of biosynthesis. These hormones are then, in many cases, transported to a site of action. So we have a component that involves transport. When this hormone arrives at the appropriate site, it needs to be perceived, it needs to be recognized to launch a signal transmission. So we have a perception site. And then when the hormone has done its action, basically it also needs to be removed, and that will fall into the category of catabolism. And these four different processes contribute to what we call homeostasis. So when we try to understand what happens when we apply a hormone biostimulant, we should be aware that this hormone biostimulant may influence any of those four processes. So it could be that the stimulant that we use stimulates the biosynthesis of a particular hormone, for example, or it could increase or block the transport of the hormone. Likewise, it can alter perception and catabolism. Now, to make it even more complicated, what we understand from plant hormone signaling today is that these different processes are also connected to each other through a feedback regulation. For example, it is well known that if we would, for example, increase the biosynthesis of hormone, we may also increase catabolism counteract this artificial increase. When we, for example, alter the transport, we can influence biosynthesis or the perception. And so, for example, it's well known that many of the biosynthesis mutants are compensated by having an increased perception. So, although we may be able to modify any of these processes, specifically through feedback, we will almost always influence the other processes as well. So, there are, however, clear examples of chemicals that can influence these processes, and I've mentioned before that it is perfectly possible to stimulate cell divisions to such an extent that we produce skulls, which is actually a group of cells that are continuously dividing and expanding, and that they result in an unorganized growth and well known as skulls. So why is this still possible? Well, it's because in this condition we are applying such high levels of hormone that we actually almost overrule this feedback regulation, and we push the system that much that we create this kind of outcome. And then the last point I want to make for this slide is that although we may think that we understand which of these processes are being influenced by the hormone biostimulant, it's also important to understand that these different processes will evolve over time and that they will be spatially coordinated, which basically means that a plant may be highly sensitive in an early stage of development, but as soon as it starts to flower, for example, it may no longer be sensitive to deregulation through a hormone biostimulant. And also it could be that specific organs or tissues are very sensitive in one moment in developmental stage, and they will not be at a different developmental stage. So then the question remains, okay, we now have seen that plant hormones can influence the cell cycle by controlling these different cell cycle phases. And when we think about how hormones can, or biostimulants can have an impact on this cell division, then we have to include these 4 different processes. Okay, so plants, they basically grow by growing at extremities. And here we see the very top of dicotyledonous plants, like an Arabidopsis model, where growth or cell division basically takes place at the very end of the tissues, and we call that the shoot apical meristem. And to coordinate that growth, and that's very important, plants have what we call an organizing center, where cell divisions are being regulated. You can actually compare cell growth, or let's say plant growth, with how a construction wall is being made. So when you have a wall of bricks, it is not possible to include a brick right in the middle of the wall. You basically have to add bricks on the top. And this is not exactly what happens in plants, but it is to some extent somewhat similar in that cell divisions are confined only to the periphery. So we have lots of cell divisions happening here at the central zone. And the more cell divisions that you will have here at the central zone, the wider the meristem gets. And the wider meristem, for example, is linked with a much larger diameter of the stem. So clearly, biomass can be controlled by controlling the number of cell divisions that will take place in the organizing center. At the periphery of the shoot apical meristem, we see this orange zone, and these are actually the sites where organs are initiated, and we call them organ primordia. In many cases, leaves are being generated, or in this case for Arabidopsis, they are actually flower buds that are being produced there. So if we want to have an increase in, let's say, leaf growth, we need to have either a more frequent initiation of organ primordia, or these primordia need to grow faster to create larger leaf blades, for example. So here again, it is the apex where decisions are made and how growth is controlled in plants. And if we would be able to modulate the processes taking place in the shoot apical meristem, we are likely going to be able to influence plant growth. So one example How this can be done is published by the research team of Thomas Schmolling, and they have been working for many years now on the role of cytokinin in plant growth, and have been focusing on a specific group of enzymes that are involved in the catabolism of cytokinin. These are the cytokinin oxidases, genes that are identified as CKX. What is interesting about their research is that they have been mutating the CKX genes, and by mutating them, basically they inactivate or reduce the capacity of the plant to remove cytokinins. And this is, for example, not shown on this slide, but basically this is well established that cytokinin oxidases can remove cytokinin by an oxidative step. So what they have found, if they make multiple mutants of CKX in Brassica napus, they observed that these plants are producing many more flower buds than the wild type condition. So this is shown in these pictures. We can see that the number of flower buds are increased. And so here you see the quantified data in a CKX35 mutant. We see that there are significantly larger number of flowers being produced by Brassica napus or rapeseed. And this translates then not only in larger numbers, but also in a higher seed yield. And basically, they have shown that by creating this mutation, you can increase the yield of Brassica napus. What's interesting is that the similar kind of CKX mutants have been already produced, also in rice and wheat and Arabidopsis, and there they find similar type of results. Why is this for us interesting? Well, although this is a genetic study, it is clear that if you would be able to inactivate those cytokinin oxidases, the CKX enzymes, if you can inhibit them, then you may actually create the same effect. And this has been already shown for a number of products. One of them is, for example, Insight. This is a product that was identified as a homologue of cytokinin, but it actually is shown to have an action as an inhibitor of CKX. And our own work, when we screened for molecules that can stimulate shoot growth, we identified phenyladenine and basically reported that this phenyladenine is also an inhibitor of CKX enzymes. And so by inhibiting the degradation of cytokinin, we can stimulate shoot growth in an in vitro condition. So here is a beautiful example how you can modulate a specific subset of hormone homeostasis and have a positive impact on crop growth and yield. Now, on the other side of the plant, we have roots, and roots, they grow at the tip. And so this is very important to understand because here a number of processes take place, which are to some extent similar to what happens in the shoot apical meristem, although there are important differences that we need to take into account. So root tips, they are organized also with a center. And here we have these blue cells, which are a group of cells that do not divide very often. They are the quiescent center cells, and they are surrounded by what we call the stem cell niche. These are cells that divide on a regular basis and create the different tissues that can be shown, that are shown here in different colors. So with the vascular tissue in the middle, the endodermis and pericycle, cortical cells, and the root cap here at the bottom. So this is this is where the cells are dividing and growing. So this is the meristematic zone. And behind that meristematic zone, we have what we call the elongation zone. And here we see that the cells are actually no longer dividing, but they are stretching. And then beyond that zone, we have the differentiation zone. And here cells are differentiating into different functions, such as, for example, the creation of root hairs. So we can distinguish different regions within a root tip, and we can say that cell divisions are primarily taking place in the meristematic zone. And we know, for example, that auxin and ABA are very important hormones to control the divisions in the meristematic zone. Here is a summary of the different types of hormones that have been shown to influence the growth of roots at the tip. Again, we have this quiescent center surrounded with the stem cell niche. SCN. And these group of cells, like I said, are controlled by ethylene and auxin, but you have also an interplay between them with ethylene and Brussels steroids. So the quiescent center doesn't divide very often, but they do contribute to maintaining the initial cells around the quiescent center, and therefore can decide how frequently new tissue layers are being established. At a little bit higher up, where you have a lot of cell proliferation still at the primary meristem, you have the same kind of hormones, including gibberellic acid, that can play a role in this cell division. And then we have the transition zone, where cells are going to start to stretch a little bit more. We have a lot of endoreplication going on, remember. So you have cell differentiation and expansion. And then you have the elongation zone, where cells really are getting a lot longer. And then here, another group of enzymes is playing a role to control these processes. So you can see this is very, very complicated. In one position, you may actually stimulate stem cell maintenance by auxin, and at another position, auxin actually counteracts endoreplication. So it's clear that if you would use a single hormone to try to manipulate plant growth, you probably are going to have multiple effects, not just increase of cell division somewhere. Now, to illustrate that a little bit more in detail, we don't only have these hormones working in different zones. In fact, what we now know is that different cell types are sensitive in different degrees to the different hormones. And for example, the endodermis is highly sensitive to gibberellic acid. The auxin is controlling the quiescent center, vascular tissue development, epidermal cells. Cytokinins are controlling vascular bundle development. Brussels steroids are influencing, the epidermal cells are very sensitive to the Brussels steroids, etc. So again, you see that the whole palette of plant hormones can actually influence different cell types at the root tip and thereby control growth. Growth of roots is not only limited to the elongation, or let's say growth of the root tip, also branching is a very important component of establishing root architecture. And so basically, what we know is that the root tip decides to prime where new lateral roots are being formed, and this process is highly influenced by auxin primarily. Although that auxin can also stimulate lateral root formation, it does play a role in the different steps during lateral root formation. So in a former study that we've conducted in our lab, we have looked at different plant extracts and looked how they can influence root tip growth and lateral root formation. And what we see here for primary root length is that the control generates a root that has a certain length, almost 4 centimeters, and that there are a bunch of plant extracts have been able to stimulate primary root growth, including Belgian endive root extract. That was one of the best stimulators. Another product which we know maybe quite well is Kelpak. This is a commercial product. Here we see that there is actually a strong inhibition of root growth. Still, Kelpak is known as a biostimulant, and we may actually explain this by the fact that Kelpak may cause some stress to plants, and under certain conditions, priming of stress can be helpful to help plants overcome stress in outdoor conditions, and that's maybe why Kelpak has shown to have positive effects on plant growth. So you see, there are a number of extracts that quite a reasonable number of extracts that can stimulate growth, and there are an equal number, a bit more, that can reduce growth. We did the same for lateral root formation, and here we see that the control is more to the left side. So there are fewer products that can really stimulate lateral root induction and formation, whereas most of the products we see that this is inhibited. Now you need to know that also primary root growth and lateral root growth are somewhat connected to each other, but here clearly we can see that we can separate to some extent the processes. So we have tested Belgian endive products, and we found that it can promote shoot and root growth in vitro, and it can stimulate, for example, root growth in Arabidopsis thaliana, but it can also stimulate the adventitious rooting in Plectranthus, which is an ornamental plant, and we have seen that the length of the roots and the number of roots have increased in vitro when we apply that to these specific ornamental species. Next to plant cell division, we have also an increase in growth by means of cell expansion. And here also plant hormones play an important role. Cell expansion is far less well understood compared to plant cell division. We know that there's an important role to play for proton pumps. These are enzymes that are located in the plasma membrane, and they will pump protons from the inside to the outside, acidifying the cell wall contact. So in the presence of oxygen, the proton pumps will be activated through a mechanism of modulation through kinases and dephosphorylation with phosphatases, which is controlled by auxin. These are auxin-regulated proteins, and also by Brussels steroids. And so basically, upon activation of proton pumping, we loosen up the cell wall, and this will allow cell expansion to take place. Cell expansion is a very important contributor to plant growth, and this can be best illustrated to look at leaf growth which is shown in a cartoon here. When leaves are initiated at the primordium, they are very small, of course, and many, or if not all, cells contribute to cell divisions. And as the leaf grows in size, fewer and fewer cells are contributing to cell division, whereas more and more cells are contributing by cell expansion. And we see that the majority of cells are basically located at the petiole side of the leaf that contribute to cell division, whereas cell expansion is happening more at the top. And when you look at the final stages, we see that the majority of increase in size is actually attributed to cell expansion rather than to cell division. So now we have handled the first group, and time is progressing quickly. So I've decided to only talk a little bit about regulation of metabolism and limit it to the fact that many studies have shown that jasmonate is an important regulator of the regulation of secondary metabolism. Many plant metabolites like terpenes, phenolic compounds, alkaloids, and glucosinolates are specialized metabolism which is controlled by jasmonic acid, for example. And to save a bit of time, I will skip to the next group of actions that we know are connected with biostimulants, and that is plant stress response. And I would like to give you two examples, one on salinity and one on heat, how biostimulants and how plants respond to these different kinds of stresses. So high salt causes a number of changes in plants, and the first step that happens is that applying sodium chloride, for example, will cause ionic stress. And this is being sensed by proteins that are located in the cell wall and in the plasma membrane. And these are proteins like these receptor-like kinases. We think that they are involved in sensing whether there is too much sodium in the cell wall present, and that will activate inside in the cell a number of regulators like SOS, which is super sensitive to sodium chloride. So these downstream regulatory signals will help to actually the plant to mitigate the salt stress by trying to increase the compartmentalization of sodium, for example, and pumping it into the vac wall. A second consequence of having salt stress, sodium chloride stress, is that it also will cause an osmotic effect. And here there are a number of other proteins that are known to be located in the plasma membrane or close to it, like these histidine protein kinases and non-selective chloride channels. These are proteins that actually are activated or sense the osmotic stress and try through a downstream MAP kinase signaling pathway to change gene expression and then activate a number of proteins that try to increase transport of ions across the membrane. And so by that, they will contribute to producing a lot more osmolytes, and that will then compensate for the increased osmotic stress. And then a third group of effects that happen upon salt stress is that we get oxidative stress, and this is the result of an increased production and accumulation of radical oxygen species. And then this will launch a number of other processes downstream. So stress proteins are being activated, and there will be activation, or let's say the ROS homeostasis will be altered in an attempt to come to salt tolerance. So it's very complicated indeed. There are 3 levels of signaling that happens after a high salt stress. And basically, we try to understand how this now works in plants and how would a biostimulant be able to help plants to mitigate this salt stress. And without that we know exactly how it works, we know that the plant cell wall plays a very important role in sensing and regulating plant cell salt stress. So one of the things that can happen is by increasing the salt content in cell walls, this can create tension signals. So that means that the position or the structure of cell wall components, which include proteins, pectin, polysaccharides, cellulose, phenolic compounds, by changing the interaction between the different components in the cell wall, we will alter the tension in the cell wall, and this can be sensed by a number of proteins, and some of the proteins that can sense mechanical properties, for example, is pheronia, but also these receptor kinases, they are there to sense it. So what we think is that by salt stress, you are changing tension, you are changing these interactions, ROS is being synthesized, you have acidification of the cell wall, and all of these processes will trigger a response, and the plant will try to help itself to mitigate that salt stress. And when we think about biostimulants, then we should think about molecules that may influence these processes that happen into the cell wall. This can be mitigation of ROS production, this could be a release of calcium, this could be changing the interactions of one of these components. So if we summarize that, we can see that biostimulants can actually influence receptors or sensors that are present in the cell wall or in the plasma membrane. So they can influence the interaction between the different components of the cell wall. And so this will have an impact on cell morphology and structure and change gene transcription, which then downstream regulates a response factor, a response regulation that can help the plant to mitigate stress. So we have actions through the cell wall or through the cell membrane. And in the cell membrane, we have a lot of transporters, we have protein kinases, we have proton pumps, we have sphingolipids, etc. And here again, biostimulants can change the action of these proteins, change the activity of ion transport, and by that, they can change the protection of the cell, change the gene transcription, and create a response. So when we think now about hormones, we see that they are far more downstream in this process compared to cell division, cell expansion. We see that ABA can, for example, change the downstream regulation of salt stress responses and modulate how plants will mitigate the stress. So we have talked a lot about different components upon salt stress, and one of them is the production of reactive oxygen species. And these are small chemicals that come in very different forms. We have hydroxyl groups, we have O2 minus, we have peroxide, but there are actually many different kinds. And when those are produced, they are actually causing a lot of damage to your plant. And this damage needs to be repaired and triggers a number of processes. This damage can be to the photosynthesis apparatus, this can be to the fatty acid beta oxidation, there can be lots of processes that are actually influenced. And one of them is, for example, also the NADPH activated pumps that will also produce oxygen radicals that creates a signal that can propagate throughout the plant. So we have damage at different levels, and many of the processes that can help plants to overcome stress will actually dampen ROS. And so we have different options. We can reduce the levels of ROS by having an enzymatic defense or a non-enzymatic defense. When we look at the enzymatic defense, there are a bunch of proteins that are being synthesized by the plants themselves that will remove these different types of ROS molecules. And upon stress, we see that many of these are being activated or being transcriptionally produced. On the other end of the spectrum, we have this non-enzymatic defense mechanism. And here we see a lot of antioxidants like vitamin C, ascorbic acid, but also glutathione, carotenoids, flavonoids, alpha-tocopherol, and many other products that we know they can quench the activity of ROS. And so we know now that biostimulants either have an antioxidant activity or they can activate enzymes that remove ROS. We have one example here in our own lab that shows that sodium chloride will reduce shoot emergence in very young seedlings of Arabidopsis. And when we apply 100 millimole of sodium chloride, we see that there is a strong decrease of growth of these little seedlings. However, when we apply an extract from sunflower barks, SBE, and this dose of 0.5 grams, we can actually partially overcome this inhibition, and we see that shoot growth is actually stimulated under these conditions. And when we look at the action of our sunflower bark extract, we can see that it actually quenches the production of H2O2. So the H2O2 levels upon cell stress will dramatically increase in the control conditions, whereas the increase in the SBE-treated plants is much lower. And this is not only shown by measuring the H2O2 content, but also using another chemical that can stain oxidative species being produced, DAB. You see the same kind of increase in these brown conditions where SBE has been applied. Okay, so this is a beautiful example how ROS is going to be mitigated by an antioxidant. Now, ROS is not only playing an important role in biostimulants that are from organic nature, also bacteria can be an important component when we talk about ROS. And here I like this paper a lot. It's a paper where they show that the interaction between a plant and a plant growth promoting bacterium, Bacillus thuringiensis, that this involves actually the increase of ROS production for a short moment during 20 minutes, and then it declines again. And this is actually the activation of, so plants recognize the bacterium, they start to produce ROS as a response to the contact with the bacterium. And then in response to that, the bacteriums will start to produce auxin, which is a plant hormone, but now being produced by this bacterium. And by doing so, it actually activates the plant to allow the bacteria to colonize. So once the bacterium has synthesized this auxin, IAA, it will stimulate the plant to allow the bacterium to colonize. And upon colonization, the plant will produce more lateral roots. So here you see how a combined modus of using the capacity of the plant to produce ROS will have an impact on the capacity of the bacterium to produce auxins, which then in turn will stimulate the plant to accept this bacterium to modulate lateral root formation. So it takes two to tango. And so I think I can still carry on. Erika, should I stop here or do you want me to carry on?
No, you can finish the presentation.
I can finish. So I have this second example, which also interests me specifically, and that is to look into the impact of temperature on crop production. High temperatures can cause severe damage, and the first impact they will have is to inhibit photosynthesis. This can be because of direct damage to proteins that are required for photosynthesis, so it damages the oxygen evolvent complex of the photosystem too, but it will also obviously have an impact on evaporation of water, and this may close stomates and prevent further exchange of gas that is required to feed the photosynthetic process. So it damages the thylakoid membranes and it will reduce rubisco activity. So these effects have a direct impact on energy metabolism, and you need to repair proteins in order to be able, for the plant to be able to continue to grow after heat stress is passed. However, I am specifically interested into a phenomenon that is also known to happen specifically with heat stress. And that is that in many crops, we have observed that the moment of reproduction, that the plants are much more sensitive to high temperature stress. And so this is shown for two crops here, for corn and soybean. So this is the optimum growth temperature for corn during the vegetative phase, and we see that the optimum is at about 80 degrees Fahrenheit. The optimum growth conditions, however, for the reproductive stage, when we are building the ears and seed production, then, or the fertility moment, we see that the optimum is 10 Fahrenheit degrees lower than the optimum for vegetative response. And similar conditions have been described for soybean. And in fact, this is a process or a phenomenon that we can generally observe. So here, this is not a problem of energy metabolism. This is something that is strictly linked to the reproductive phase, where you have different cell types that need to be developed, and they are sensitive to high temperature. Now, what's also very important that you will see that the optimum for soybean and corn are more or less the same, but it's clear that the sensitivity of, let's say, the optimum temperature can vary profoundly for the different crop species that we are looking at. So when we were looking at tomato, which grows at a little bit lower temperature, we found that during the production season in 2019, there was damage because of 3 consecutive heat waves that were recorded in Belgian summer. So here what we see is the production in a regular greenhouse, that the oldest fruits that are flowering first, they are bigger, and then when you go to the top, that the fruits are a bit smaller because they have been generated a little bit later. However, when we look at the fruits that are developed under conditions of heat stress, we saw that the oldest fruits are actually much smaller compared to the younger fruits. So basically what happened is that the heat wave hit this fruit, this flower, much more, and this one was being produced under a moment that the conditions were more favorable. And you can see the impact that it has on fruit development, which is a consequence of reproductive phase, whereas the sepals, they are very large, so they were not suffering from reduced growth. So here you see the sepals reaching the rim of the fruit, whereas in this case, the sepals are far outreaching the size of the fruit. So the vegetative growth was clearly not impacted by this heat wave. So why is that? Well, basically, there are many, many underlying processes that can influence the fertility of flower. And one example here is that under high temperature, the flowers of tomato will increase the length of the pistil. And when this is done, the fertilization of the pistil is reduced because it is an auto-fertilization process, but you need to shake the flowers a bit. And when you have this situation, you have reduced fertility, and that will create less seeds, and they will then not support the fruit growth so much. In Arabidopsis, we have a different situation where high temperature will reduce the filament size of the anthers, and this has also a consequence for fertility. So what we have learned in the past is that there are many examples, in fact, where plant hormone application can restore high temperature stress that is applied during reproductive phase. And here is one case for barley, where application of indolacetic acid, auxin, can actually restore the fertility in barley. So this is a beautiful example where biostimulants that contain plant hormones, such as, for example, auxin, may be applied to increase the fertility upon heat stress. With this last example, I would like to close my webinar and acknowledge my entire Horticell team for creating a very enthusiastic environment to do research and to participate to this highly interesting topic of biostimulants. And I thank you for your attention.
Thank you very much for this very interesting presentation and for showing the interaction between biostimulants and plant hormones. Now we will have a quick Q&A section. I just want to remind that is still possible to submit your questions using the Q&A box at the bottom. We have already received a couple of questions, so I will start immediately with the first one. Are plant hormones currently used as biostimulants? And I will add also another similar questions, which is several hormones are registered in Europe as PPPs. So could these products be sold in Europe as biostimulants instead?
So let's start with the first question. Are there already biostimulants containing hormones? Yes, and they are sold. And in some cases, it's even so that a pure plant hormone is being applied for specific processes to be modulated in plants. One of them that I know is the use of abscisic acid to alter the colors of grapes. So there are many grapes that do not color it sufficiently intense in accordance to the public or let's say the consumers' demands. And so this can be done by spraying with ABA, but the same, to stay in the same crop production system, we know that we can apply gibberellic acids to create seedless grapes, for example. That has also been used, maybe not in Europe, but definitely in the US and Israel. This is being used to modulate properties of plants in a very specific way. And what you will see is that in many cases, when they use plant hormones as the active ingredient, that they will apply that at a very specific moment during the production cycle, and usually towards the end, so that there is no inadvertent side effect that you can expect. So when the fruits need to be treated for a while before you harvest them, you don't worry too much about, for example, impact on other processes that may not be so important anymore. Now, to answer that next question, whether they could be sold as biostimulants, so this is something that the regulator will need to decide. So what I understand is that whenever you take any plant extract, there is a possibility that it will contain plant hormones, especially those hormones that are highly stable will be present in your extract. So what I also understand is that in many cases, some of the activities that we see are attributed to these hormones to be present. So in my opinion, it is possible, and depending on how you describe your product, you need to, for example, describe the content. I guess that if your product contains more than 1% of a hormone, that you will need to describe that. And then it's up to the legislator to say whether or not they can accept it. I cannot conclude whether they will accept it or not.
Okay, thank you. We have another question. So thank you for the nice presentation. And based on your experience, which types of hormones are mainly affected by biostimulant substances?
So I do not know, because I don't have a clear overview of the different biostimulants that have been tested to see how hormone signaling is being affected. I think we are at the beginning of studies looking into how hormone signaling is being affected by biostimulants. But I have no doubt that biostimulants will do this. So I think I tried to explain that we have these three groups of activities. And I think that, for example, if we would see that a particular biostimulant would enhance shoot growth, then I think it is very likely that it will affect cytokinin levels, for example. How it will do that? Well, there are many ways that it can happen. But to answer which of these hormones specifically are more affected by biostimulants, I do not dare to answer that question. I think we do not really know the answer to this question.
Okay, thank you. Next question. So the question is it preferred to have hormones into biostimulants or molecules that will produce hormones after using in plant metabolism?
Yeah, I think that's an interesting question. Yes, indeed. So what I've learned from chemical screens that have been used in molecular studies to look into cell growth and expansion processes, that in many cases they find molecules that are seen or that finally are identified as what we call precursors. So molecules that as such do not have the same chemical structure as plant hormones, but after metabolization they become a plant hormone. So the advantage of such molecules is that, for example, the plant can initially not attack these hormones because plants, they have learned to live in an environment where bacteria and fungi are able to produce plant hormones in order to change the behavior of the plant such that they can now attack the plant. In other cases, like the plant growth promoting bacteria or organisms, this can also be specifically to help the plant. So there's many options there. So if you have a precursor, this precursor needs to be converted, and that actually can alter the response to these applied products. And they may be actually accumulating to higher levels before the plant starts to dampen the effect. So I think that's of interest to use precursors. The other thing is, of course, that a precursor can also be modified in such a way that it is more soluble, more stable to light conditions, UV and sunlight, so that you can spray it without having it broken down. We know that many oxidants, for example, are sensitive to light. So you can modulate the property of that chemical so that it is better introduced into the plant and can cause its effects. So I would prefer a precursor in many cases. And maybe one last comment that I would like to add here is that plants, so for example, what is a surprise to me, that when you apply a certain hormone to a crop, for example, and not only Arabidopsis, in many cases you will hardly see any effect. You need to use very, very high doses for it to really cause an effect. And I think that's because plants have found a way to absorb shocks like production from a bacterium so that they can, when this hormone comes in, they immediately can neutralize this effect. Now, what they cannot neutralize so easily is when you, for example, affect the catabolism of hormones. So I gave you specifically that example because I like it. So if you would inhibit the CKX enzymes, and then the plant cannot, the only thing it can do is start to produce less cytokinin, in fact, which ultimately it will do. But if you completely block all the breakdown of the hormone in cases of cytokinin and auxin, etc., then plants really, really will respond. Okay, so I think looking into the catabolism side is much more interesting than looking into, for example, changing the biosynthesis directly.
Good. We have two more questions. The first one, there is a common belief that some commercial biostimulants like seaweed are acting by supplying hormones to the plants, while others say then they only modify the hormones homeostasis. Which is your opinion?
Yeah, like I said, I think if you apply hormones, so seaweeds have similar kinds of hormones than higher plants. So if you apply hormones directly to a plant, you will often see no effect. So for example, you can spray your plants with cytokinin and you hardly see an effect. Basically, the few examples that we see from the past that we can find in textbooks, they have sprayed really a lot of these hormones to get the effect that they show on these pictures. But I've tried it myself, and actually I was surprised that I saw nothing. So I think that the hormones that are present in seaweed are not likely going to be causing the phenotypes that we see, although I cannot exclude that to some extent they contribute to the effect. However, I do believe that seaweed do have molecules that can alter the context, for example, in cell walls, that will influence a ROS production or anything else, that then have a secondary effect downstream in changing hormone signaling. So I would be more inclined to think that plant extracts and likewise seaweed extract work mainly by other molecules than the hormones that they contain.
Thank you. One more questions before closing the webinar. So many thanks for your presentation. What do you think about hormone-like peptides?
Yes, so I think they are highly interesting because peptides are unlikely to be very toxic to our environment. So, you know, some of the secondary metabolites we can find in plants are extremely toxic. So if we would start using them in high amounts, that might be a problem. Whereas I think peptides are not very likely to cause so much damage. So we are at the beginning to understand about plant hormone, let's say plant peptides. So there have a number of them have been identified. And in some cases, they are just extremely specific for a specific crop. And the one example that I know is histamine from tomato. And this histamine occurs only in tomato, and you can use it to help the plant to resist better abiotic stresses. So that would be a downside, because if individual plants or species have developed their own unique peptides, then we would need to study for every crop which peptides they actually are using. So that might limit the potential of these peptide-like hormones. But other than that, I think they are there. They are most likely used by the plant to communicate between different tissues, how to respond to an environmental condition. But we are not near to understanding how they work and what their sequences are, etc. We need a lot of research there, I think.
So thank you very much for your answers. This was the last question for today. I would like to thank you again for the nice presentation of today. And I would like also to thank everybody for attending the webinar. And if you still have questions, you can submit them on biostimulant.com website, where there is a specific section called Ask the Expert, and our scientific committee will get back to you with the answers. I would like to remind you that the recorded version of this webinar will be available soon on biostimulant.com and on our social media in the next days. So stay tuned and have a nice day, everybody. And I hope to see you on the next webinar. Bye.
Bye-bye.